And so to start, it would be good to look at the history and of the success of biostatistics in the last 60 years. So there are two things which are important. The first is the biostatisticians have been involved in cutting-edge research, and secondly, the biostatisticians have led the scientific collaboration drive the methodology development. And by building such a bridge between quantitative science and health science, then we are able to help develop better research and also develop better prevention and the treatment strategies and also better health. So now we are moving into a different world, and with those massive data many of the speakers have described in biomedical science, and I use 3 examples here. One is the genome, and in particular I'll discuss the whole genome sequencing data. Secondly is exposome. So in the old traditional epidemiological study, one measures a small number of exposures, but now one can measure spectral of exposure, for example, a large number of metals, and or the air pollution measure using the satellite. And the third is a massive phenotype, and for example, like electronic chromatic record. So here are the 3 examples. And so one is the whole genome sequencing data, and then the second is the smartphone data, and so which can be used to measure all kinds of exposure information and also the phenotype information. And the third is the electronic chromatic record. And so I'm going to use those 3 examples to illustrate the importance of collaboration across different quantitative disciplines. And so the value of big data, as many of us know, is not how big the data are, is the analytics. So in particular, and the core of the data science involves 3 pillars. And one is the statistics, and the computer science, and also the informatics. So they, as you can see, this really involve a Venn diagram. So for example, at the interface of statistics and the computer science is like machine learning, for example. And so what is the goal? I think several speakers mentioned this. The goal is really the inference. So in order to transform the data into knowledge, inference play a critical role, as Todd mentioned, how create the error bars and to assess uncertainty, how can one make the hypothesis testing, how can one make a prediction to account for the uncertainty. And so to illustrate how the field has evolved, I just use my own department as an example. About 10 years ago, in my own department, we have about 30 faculty, and almost everybody has a degree in statistics or biostatistics. 10 years later, right now we have about 28 primary faculties, and you can see about a quarter of them have degree not in statistics and in like computational biology, computer science, physics, or evolutionary biology. The reason this happened is because of the need of scientific field. And so we really need this kind of interplay of different quantitative science to really solve the big biomedical problems. So this introduces the concept of the connected health. So the one component of that is the clinical data, such as EMR data. Second component is the genomics data, such as the whole genome sequencing data, or the smartphone data that can measure both the phenotype and also the exposure information, and lifestyle data, and also environmental data. So the idea is we have this different type of data and put this into the data science wonderland, and hopefully that by crunching the machine, and we can help, that can help us to understand the causes of the human diseases, and also develop a precision medicine and prevention strategies, and also better health policies. So now I'm going to illustrate using the 3 examples. The first one is the genome. So I'm going to focus on the whole genome sequencing effort. So for each human genome, any of us, and the whole genome, human genome consists of the 3 billion base pairs. So basically, if you think about the data, and they are, for each person has 3 billion columns maximum, and the letters are TCGAs. And so many of you have heard about the GWAS study. So GWAS study only occupy about 10% of genome. So you can see that GWAS study has been successful in identifying the relationship between the common variants and the human disease and the traits, but it missed 90% of the genome. By doing the whole genome sequencing, that will cover the 100% of genome. That basically is the whole human genetics. After we do the whole genome sequencing, and that's it, that whole genetics code, no more. And so 90% of the genome occupies the rare variants. And so that makes the analysis much more challenging. And to illustrate the human whole genome sequencing timeline, so about 6 years ago, maybe 8 years ago, and that was the launch of 1,000 genome project that basically sequenced about 1,000 people. And last year, was the launch of this TOP-MAD initiative by NHGRI. So that stands for Transomic Precision Medicine by the Heart and Lung Institute. So NHGRI plan to do whole genome sequencing of 100,000 to 150,000 people. And so right now they have finished about 60,000. So the informatics center is located in Michigan in the biostat department and PI'd by Gonzalo. And so another initiative is called the Genome Sequencing Program, and so that is by NHGRI. So the plan is to do whole genome sequencing of 200,000 people. By combining the TOP-MAD and GSP, that will have about 300,000 to 350,000 people with the whole genome sequencing data. So after this is accomplished, and this will give the largest number of people in the whole world, and in the world with the whole genome sequencing data. And so by looking at the preliminary data which has been generated in January, about 15,000 people. And so you can see the data are very, very sparse. So among the totally about 159 million variants has been identified. Among them, 44% of the variants are singletons. So that means if you think about the data matrix is 15,000 rows and 150 million columns. Then about 44%, that is about 60 million columns, and each column has only one, all the rest are 0. So those are the private variants. So that's why all of us are very different from each other. And so then about 14%, 90 millions, and columns had only 1 or 2, all the rest are 0. So the data matrix are very, very sparse. So that makes the inference very difficult. And so NHGRI found the genome sequencing program. So, so it found about 4 complex disease centers and 4 Mendelian disease centers. And also, NHGRI recognized the importance of an analysis. And so NHGRI found 3 analysis centers, and we are one of the 3. The other 2 are Stanford and Vanderbilt. So what is the first goal? The first goal of the whole genome sequencing analysis is sparse signal detection. Basically, one should look for the needles in the haystack. So if you look at this gentleman, he's holding a 3 billion pieces box. That is a whole genome with 3 billion base pairs. And this gentleman said, I think I found a corner piece. So that is the genetic mutation caused breast cancer. Okay, so the question is, how can we find the corner piece? And there are multiple corner pieces. So that will involve interdisciplinary strategy, involve statistical strategy, computational strategy, and also biological strategy. So the team have to really work together. So the first goal of the whole genome sequencing is scan the whole genome. Try to identify the regions which are associated with the human diseases. And so if one think about analytic challenge, right now the challenge really not how many people we can sequence and how much data we can generate. The challenge is how we can analyze the data. So about 10 years ago, and when the microarray technology just came out, and many of us, we talk about small n, large P problem. So that is about 1,000 people with the microarray data and then 20,000 genes. And 10 years later, and with the whole genome sequencing effort, so what we are dealing with is a large and huge P problem. So for example, in the GSP, and N is 200,000, the P is up to 3 billion. And so this also, when the sample size increases, more and more real variance will be observed. So this is the perfect example that the number of variables, number of variance in this case, increase with the sample size. And so because of huge data, so the method, in order for the method to be practically applicable, and the method has to be scalable, and that can handle the large data, analysis of large data. So what are the specific analytic challenges? So the first is the analysis units are not well defined, and so for the whole genome involve a large number of non-coding regions. And also biological signals generally are weak and sparse, and one need to think about the inference and to test for high-dimensional weak and sparse alternatives. And also, in order to improve analysis power, and that is where the biology come in. So one need to incorporate functional information, for example, from the encode to boost analysis power. So for the variants which are more functional, we weight them more. And so what that tells us is the statistician and the computational biologists really have to work together and to analyze, to successfully analyze the whole genome sequencing data. So as a statistician, and then it says, to save time, let's just assume I'm never wrong. And the computational biologists, they calculate the wonder of land to understand the biology. And to show you how the data look like, and so here is about 500 people, and with 100 SNP. And so totally the bright dots indicate 1 or 2. The black dots indicate 0s. So if I shuffle the subject, you can see in each column there are lots of blacks, so lots of 0, only a small number of 1s. So the data matrix are very, very sparse. And so here is the challenge. Because the data are many variants are real variants, so we cannot analyze one SNP at a time, one variable at a time, and so we really need to analyze one region at a time. So, for example, if the variants are too rare, if one can even not fit the logistic regression. So how can we perform the region-based analysis? So here's one example. Suppose this is one gene, and suppose there are 100 variants here. Suppose there are only 4 signals here. So ideally, what we want to do is we want to see whether this region is associated with the disease. But I don't want to do 100-degree freedom test because it loses power. Ideally, what I want to do is, if God tells me there are 4 signals, I take these 4 signals out and do 4-degree freedom test, right? But reality is, I don't know whether they are 4. I don't know where they are. So this strategy doesn't work, right? And also, estimation in this problem is very difficult because the variants are too rare. So now how can we do that? So that's why the inference kick in, statistician can help in this context. So I want to test this region with 100 variants associated with disease or not. I don't want to do 100-degree freedom tests. If I know the region has lots of signals, a lot of those betas not equal to 0, and in this situation, this is SCAT procedure and developed by my former student and postdocs, and Singer is in the audience here. And so this has been quite heavily used in the whole genome sequencing analysis. This strategy worked quite well. But if the signals are sparse, and suppose in this situation only 2 variants are causal variants, in this this SCAT strategy will not work well. And so two of my former students, they developed this we call the generalized hierarchism approach. So that will be more powerful to be used to detect the sparse alternative. So we apply this generalized hierarchism strategy to the breast cancer GWAS study, which has about 1,000 cases, 1,000 controls. We did the gene level analysis, and so there are multiple genes which are well known in the GWAS, in the breast cancer literature pop up. For example, FGFR2. So now let me move on to smartphone and the data. So to illustrate, I'm going to use one example. This is the work of my colleague, G.P. Alnila, who's a computer scientist that in collaboration with my colleague, Victor Di Grutola, who's a biostatistician. So here's the idea. So we are interested using the cell phone data to construct social network. So for example, if Al and I, we call each other 3 times, and then our edge will have a count 3. So that means 2 of us, we know each other, okay? And through this cell phone calls, one can construct this social network. For example, like here, and there are three networks here, and then three hubs. Why this is useful? The reason is, if we can apply this strategy in HIV prevention, and then that will deliver a more effective HIV prevention strategies. To show you how. Now think about the traditional way. For traditional way, and here, so suppose we only sample a small number of people, we do the random sample. Then what will happen is most likely the random sample here will not be the hubs, right? Then we deliver the intervention, and then you can see lots of people around the hub are affected, okay? But now let's think about different strategy. We are going to network-based sampling. So instead of doing the random sample, and what we are going to do is we are going to study the network first. After we study the network, we identify where the hubs are. We are going to intervene the hubs. Then what will happen, you can see lots of people around the hub, they are not infected. Okay, so what this tells us is if we have collaboration between statisticians and also computer scientists, this can really generate powerful new ideas for intervention. This also presents the challenges about analysis, about the statistical inference. For example, how can we do designs in this situation? How can we design network-based study? How can we know the random sampling is not effective, but if I want to do randomized clinical trial based on network, and how can we do random sampling? And how can we avoid bias sampling? And also, in causal inference, subjects are independent to each other. But this is not true in infectious diseases because the subjects are related to each other. Then the traditional assumption about independence among subjects is violated. So one needs a causal inference strategy to account for interference, the contamination. Okay? So this is where the statistician can help. Now let me move on to the EMR. So EMR data are rich, but they are messy. They are very noisy. And okay, I think you all know better than I do. Okay, so the EMR data, generally we know we use ICD-9 code, right? And to construct the phenotype information. But if you think about ICD-9 code, those are constructed for billing purposes, right? And so therefore, the data, if they are not used to design for research purpose, the data are noisy. So in order to get more accurate data, and then we need to incorporate unstructured data. Those are the doctor notes. So if one can use a natural language processing to mine the text mining, the doctor's notes, and then combine those data into those data matrices we are more familiar with, and then that will create a better data. The second challenge for the EMR data is there are lots of missing data. Okay, so how can one handle missing data? So here is why we need to use the unstructured data to improve the data quality. So here is a positive predictive value of pneumoroid arthritis. So if one used ICD-9 code alone, and then if one calculates true RA probability, it's about 56%. But if one uses ST9 code plus natural language processing to mine the doctor's notes, you can see the positive predictive value is 94%. So what that tells us, if with the collaboration of biostatistician and the medical informatician, and we can really get more accurate data. So just a few final words and to move on with the data science in the health. And so we need to work together to solve big problems. So the statistician, computer scientists, and informatician and domain scientists really need to work together, and then we can solve big problems. Secondly, we have to really think about how to promote study design. Bob gave an excellent talk this morning about the importance of study design. Just think about the election this year. The poll results are completely wrong. And so how can one design better study to get a better polls? And also, how can one handle the non-response bias? And also, more effort needs to be put on inference for big data. And also, how can we build a better infrastructure, in particularly, and the field is moving in the cloud environment. For example, the GSP and also the TopMed for the whole genome sequencing data, the data are too big. So the traditional way, put the data in dbGaT, have each individual investigator copy the data to their own cluster with the resources. Better to do it, just put in the cloud, and everybody access the same data. And finally, encourage the data sharing and also the software sharing. Yeah, thank you.
The patients must satisfy that medical home definition for the first time between July of 2010 and March 2013, and then we follow them until July of 2014. So that's sort of that defines our cohorts.